So please mute your mics. Thank you. So today we are going to talk about two things. One is how to map genes on chromosomes. And then we are also going to talk about sex-linked inheritance or sex chromosomes, how they were discovered. But before we get into details of today's lecture, let me recap what we learned last time. So last time we talked about discovery of chromosomal theory of inheritance, and what we discussed was a very important question which Morgan's lab came across, and the question was, what happens if genes reside on, if they reside on independent chromosomes or if they reside on the same chromosomes? So if they reside on independent chromosomes, according to Mendel's law of independent assortment, you get 25% each gametes, and there will be, equal parental and non-parental. So it will be 1:1:1:1 or 1/4, or you can also say 25% each. And what Morgan's lab discovered that, the factors which Mendel talked about are actually genes which reside on chromosomes. So if they reside on same chromosomes and the inheritance of chromosomes and these traits is independent during meiosis, independent of each other, so we should get, if they reside on same chromosome, we should get 50% each, parentals only, and we should get no non-parentals. This is what the question was. According to Mendel, we studied this, that Mendel was lucky. He studied 7 traits, and each of this trait was actually on independent chromosome in P's. What happens if Mendel or we study 8th trait, which means, because here P's, they have 7 chromosomes, and each of these traits, they were on independent chromosomes. So what happens if we have an 8th trait, or Mendel would have studied 8th trait? So would he have, still receive this 25% each, or 50%, this 50% each if they reside on same chromosome? That was the question. And last time we saw that this conflict for a short time between Mendelian law of independent assortment and chromosomal theory of inheritance, that was resolved by Morgan's lab by famous experiments which were done by Sturtevant. We took example of black body color gene and vestigial gene. Vestigial phenotype was this, very deformed wings, and normal body color or wild type body color is brown. So the flies they received, which were black in color, when they set up a cross between black vestigial homozygous recessive with wild type flies, in heterozygous they received all normal flies. And when they set up test cross, so test cross was homozygous recessive crossed with F1, And if they reside on independent chromosomes, we expect, you know, plus plus. So this for black, this for vestigial plus plus gamete, and then black vestigial, this is the other parental. Remember, parentals are going to be always original parents, not F1. And then non-parentals were B plus and vestigial plus, which was neither in this parent nor in that parent, which is due to independent assortment. So if they reside on same chromosome, independent chromosome, this is what we expect, 25% each. And if they reside on same chromosome like this, and the other parent is plus plus, you cross. So in this case, during gamete formation, so these chromosomes, homologs, they will go to opposite poles. In F1, for example, you get plus plus and black vestigial. You set up a test cross, and test cross is again homozygous recessive, black vestigial. So we forget about the tester parent, this one. Tester is basically homozygous recessive. We are going to look at the inheritance of wild type trait as well as the mutant ones through gametogenesis. Yeah. And what we are going to look at is the gametic combination. And in case of gametic combination, this is what they were expecting. If they are on independent chromosomes, so they will, this chromosome during gamete should go to one gamete, this one to another. So we should get 50-50 parental trait should. So this plus plus is actually coming from this parent, plus plus. Okay, this black vestigial is coming from this parent, and you know it should remain 50-50, and no non-parental should be obtained. But what they discovered was, you know, these parentals were quite big in number, 1:1 ratio, and then non-parentals were also 1:1 ratio, but very few numbers, neither 25% each nor absolutely absent. And based on that, what Morgan lab proposed, that this, we calculated this last time, that this percentage of new combinations, which today we know as recombinants, they are actually referring to the physical distance between two genes. So black and vestigial, this percentage of recombination is dependent on this distance, and they name this distance as centimorgans. So out of 100, if there are 100 crossover gametes, okay, so if there are 100 crossovers in meiosis, or let's say if there are 100 meiotic events, so if there are 100 meiotic events and we see 1% recombination, it means the physical distance between black and vestigial is 1 centimorgan. So 17, but what we receive 17%, it means, so black and vestigial are 17 centimorgans apart. If there will be 100 meiotic events, there will be only 17 recombinants we will find. That's what we refer to as frequency of recombination or percent recombination or MAB distance. So this centimorgan is actually the MAB distance. Okay. Then So this is what we saw last time. Now, yeah, we talked about maize chromosome, that the proof of recombination, it came from uh, later on from Barbara McClintock while she was working with Harriet Creighton in the maize, and I explained you while they were using waxy and starchy, two traits. The ninth chromosome of maize has this morphological uh, extension, and they could see due to recombination after test cross, they could see, they could identify under microscope this chromosome, and they could show physical basis of uh, recombination. So that is what was due to meiosis, and I would like you to I write here a question. So you have used, so and you have to bring answer to this question. You have to submit this by next lecture. So you have used, let's say, in fruit flies, you have used two traits. One is wingless, and the other is uh, wingless, and the other is, let's say, legless. Two hypothetical traits. You crossed wild type with wingless, legless flies. You received all wild type. You set up a test cross. Okay. Here you used female, here you use male. And you received, parentals, non-parentals, let's say 150, 160, 1500, and 1450. This is problem number 1. Okay. Now you used same phenotype. You crossed wild type with wingless, legless. In F1, you received all wild type. You set up test cross, and here test female, and this one F1 you used male. Here you used female, and now you go to, and what you found is parentals and non-parentals, and you found, let's say, 1500, 1550, and 00. Same traits. Question is, what went wrong? Or how do you explain? Here you calculate the distance, calculate distance between wingless and your legless genes. So that is what you should try to solve. Now, last time we also talked about cis and trans, so I'm not going to go into detail what these are. Today we are going to talk about now 3-point test cross. Okay. Last time somebody asked me a question: what will happen if we have 3 genes? If we start analyzing 3 traits, will we get parentals, non-parentals, etc.? Now, the question in front of you is, you are actually studying 3 different traits at the same time. For example, black, cinnabar, and vestigial. This is a test cross, okay? So you must have used parents. And now, after test cross, what you receive is you already know black and vestigial are 17 map units apart, and black and cinnabar are 7 map units apart. From a dihybrid cross between black vestigial or black cinnabar, these are the distances you already know. However, you want to now see how black, vestigial, and cinnabar are organized on chromosome. One possibility is this. The other possibility is black, cinnabar, vestigial, or cinnabar, black, vestigial. So here are the possibilities: black, cinnabar, vestigial, cinnabar, black, vestigial. Okay, because on the basis of dihybrid cross, these are the distances, and remember, these physical distances are additive. So you can, black and cinnabar are 7 more map units apart. Cinnabar and vestigial are 10 map units apart. So you have total 17, which is between black and vestigial. Black and vestigial. The other possibility is black is in the middle. Cinnabar, black is 7, we know. And black, vestigial are 17. Black is in the middle. So now the additive between cinnabar and vestigial is 24. One possibility is you now do cinnabar vestigial dihybrid cross and try to see whether the distance is fit, but that will even further confuse you. So we go for 3-point test cross, where we have all 3 traits being analyzed together. And after the test cross, what we receive that cinnabar vestigial is 10 centimorgans or map units apart. However, in this progeny, we also receive strange recombinants, which we call, and these numbers are less than 1% of the total. And these are basically the double crossovers. These are the least number in a 3-point test cross, and these are due to double crossover. Okay, so we have to resolve this. So this is again telling you, if you are studying 2 traits, A and B, they are 5 map units apart. A and C are 3 map units apart. But if you have to now map all 3 on the chromosome, either this picture can be true or this one can be true. So we have to resolve this. How we do? So after 3-point test cross, we take all the combinations and then we look at their numbers, what we receive. Okay. So for this, I take example from your textbook, the Griffiths. So here you have 3 traits: cross-veinless, cut, and vermilion. And here V plus means this is wild type. Okay. Cross-veinless homozygous means this is homozygous recessive, and cut homozygous recessive. If they are, let's say, V plus, it means it's wild type. If it is V alone, it's a recessive allele. It's a mutant allele. If it's CT plus, it means it's wild type for cut. And if it is CT, it's mutant allele. Cut, mutant allele. And same is for cross-veinless. Okay, this is mutant allele. And CV plus means wild type. Now you get the F1 trihybrid. F1 trihybrid will be heterozygous, which will be all normal because all these traits are recessive. Now you set up a 3-point test cross, which means you take F1 parent, this one F1 female crossed with tester male, this one, and you will get such classes like you get here. How many classes of gametes you will get? 8. You can count them: 1, 2, 3, 4, 5, 6, 7, 8. Now, such are the numbers you receive. Okay. You have vermilion cross-veinless cut, vermilion cross-veinless cut. This is vermilion mutant. This is wild type. This is vermilion wild type. These are cross-veinless cut. Okay, and you get 8 different classes: 1, 2, 3, 4, 5, 6, 7, and 8. These are the numbers you receive: the biggest numbers and the smallest numbers. If you pay attention to parentals, what were parentals? Wild type, cross-veinless, cut. These are the parentals. Parent 1 and parent 2 was cross-veinless wild type, cut wild type. So cross-veinless wild type and cut wild type. This is parent 1, cross-veinless and cut vermilion wild type. These are parent 2. Remember always, the biggest numbers you will get, I explained you last time as well, the biggest numbers you get, they are always parentals. And the least numbers, which are less than 1%, these are double crossovers. Double crossovers. These are recombinants. Now, what are the other classes? So you have resolved 2 parentals and double crossovers. Your question you are dealing with is how vermilion, cross-veinless, and cut, they are organized on the chromosome. What is the physical distance between each of these traits? Okay, you already, let's say, through dihybrid crosses you can receive them, but you will never know in a dihybrid cross which one lies in the center. It's the cut which lies in the center, it's the cross-veinless, or is it the vermilion which is in the center? So for that, we use double crossover, and double crossovers, they help us in determining the gene order, which one is in the middle. So what do you have here? For example, you have parental is what we use parentals, vermilion, cross veinless, and cut. Okay, plus and plus. And the other parental was vermilion plus, cross veinless mutant, and cut mutant. Now double crossover means it goes like this: vermilion, cross veinless, cut, and reciprocal crossover will be vermilion, wild type, cut, wild type, and this. So you will get a product of this will be vermilion plus, cross veinless plus, and then cut. Vermilion plus, cross veinless plus, cut. And the other one will be vermilion, cross veinless, and cut plus. Vermilion, cross veinless, and cut plus. Now, are these like this? In the double crossover, what we are getting is vermilion, cross veinless plus, and cut. It does not hold true if we assume cross veinless in the center. And the other double crossover reciprocal is vermilion plus, cross veinless minus, cross veinless plus. So it also is telling us this organization is wrong. Other possibility is we assume now, for example, vermilion, cut in the middle, cross veinless out. Parentals were vermilion, cut plus, cross veinless plus, and vermilion plus. Other parent was vermilion plus, cross veinless, and cut mutant. Now, if we make the double crossover, it will be vermilion, cross veinless minus, and CV plus. Vermilion mutant and CV plus, which is, so I write it here, vermilion, cross veinless minus, cut plus. And the other will be vermilion plus, cross veinless plus, and CT minus. So vermilion plus, cut plus, and CT minus. Vermilion, CV plus, vermilion, what did I wrong do?
So ये second parent CV plus होगा, आपने CT plus लिखा है.
सर CV को CTs में एक मिस्टेक हो गई है.
मैंने कट इधर प्लस करना था, मैंने यहाँ पे कट करना था और यहाँ पे CV प्लस था. या सो वर्मिलियन ये कट होना चाहिए यहाँ पे, और वर्मिलियन कट एंड देन CV प्लस, या ये CV प्लस है. सो वर्मिलियन cut minus and CV plus. So it means this order is correct. Cut is in the middle because the gametic combination of double crossovers is giving us the right combinations if this is what we assume. So this is not the only way you discover the order. There are other ways as well, other methods as well. Now, we already know the distance between vermilion and cut and cross veinless, etc. But in 3-point test cross, we get better and precise idea. How do we do that? Now, based on these gametes, we are going to deal with two traits or two genes at a time. For example, we will look at just recombinants between vermilion and cross-veinless at a given time. We will forget about cut here. So we will not worry about cut. We know already what are the parentals. So we will find the recombinant classes between vermilion and cross-veinless. And anything which is non-parental, anything which deviates from parental gametes, that is basically recombinant between vermilion and cross-veinless. That is, vermilion and cross-veinless, they were always trans. So vermilion was coming as mutant from one and cross-veinless from other. Vermilion was wild type and cross-veinless. Vermilion wild type and cross-veinless wild type, they were never in the parental gametes. So this is parental vermilion and cross-veinless. We are not focusing on, we are ignoring cut at this moment. Just focus on these two traits. This is a recombinant class, so you will write an R in front of this gamete under vermilion and cross-veinless. Vermilion and cross-veinless together, this is also recombinant because in original parents they were not together. Then vermilion and cross-veinless mutant again is a recombinant, and vermilion and cross-veinless plus is again a recombinant. Now you look at vermilion and cut. Now it means you forget about the cross-veinless. Vermilion and cut, they were also in trans. They were never wild type together. Vermilion was wild type, cut was mutant. Vermilion was mutant, cut was wild type. So you look at vermilion and cut. So this is a recombinant class. Vermilion and cut, wild type. This is a recombinant class. Vermilion cut, mutant, wild type class, and so on, the double crossovers. You add the recombinant classes, recombinant numbers of each class. Same you will do between cross-veinless and cut. You ignore now vermilion and you will find 4 recombinant classes, 93. Now, if you want to determine the distance between, let's say, vermilion and cross-veinless, you will simply take the recombinant classes, which are 268, divided by total, which is 1448, into 100, that will give you percent recombination. So you calculate, and this is what you get: 13.2 map unit between vermilion and cut and cross-veinless 6.4, and you already determined order through double crossover. You have already known this, so you will say, okay, cut is in the middle, and that's how the gene organization on chromosomes is. So there are 3 simple principles, remember, for 3-point test cross. Always we know the recombination is reciprocal, so you have to group the recombinant classes into reciprocal pairs. Most frequent pairs are parentals. We saw this. Most frequent ones are parentals. We also saw even in these examples. Here you see most frequent numbers are parentals. Okay. So and you drive the gene order from the least frequent pair, which is basically double crossover. Okay, that's how you resolve it. So when you draw a genetic map, we just saw, for example, vermilion, cut, cross, veinless. Remember, the physical length of genetic interval is proportional to the frequency of crossover. Bigger the distance between two genes, higher the frequency of crossover. Smaller the distance, lower the frequency of recombination. And distances are approximately additive. For example, if we have found cross, veinless, and cut are let's say this was 13 and this was 13.8 and this was 7.5, they are approximately additive. Vermilion and cross veinless maybe is 21 or 20 to 21 centimorgans, whatever. Okay, and then you apply stats to make sure that these distances are correct. However, if the distance normally, practically, when we perform such experiments, even between two traits, let's say we are focusing just on vermilion and cross veinless, if the distance is less than 10 centimorgans, we find very few recombinants. It's very difficult. If we go less than 10 centimorgans, recombination between two genes is very little. And I would say genes less than 10 centimorgans, you will call them linked. Genes on same chromosome, they are normally referred to as genes are linked, but true linkage is when two genes always inherit together during meiosis. No recombination. This shows zero recombination. Also, another point I would like to mention is in Mendel, for example, you received non-parental and parentals. If you remember, non-parentals were 50% and parentals were 50%. You remember they were all 25% each. If you add them together, they make 50% and they make 50% as well. If the two genes, if this is a centromere, if the two genes are on same chromosome, but in different arms of same chromosome, then we will be confused. Then we will get, such 50-50. Because the distance is so big, spanning over centromere, that recombination will show you, that they are on independent chromosome. But once you use another trait, let's say you use A and C, and A and D, B and C, B and D crosses in dihybrid, only then you will realize that A and B are actually on the same chromosome, because C is on same chromosome, D is on same chromosome. This was simple thing, but remember, crossover can be at multiple positions. There can be multiple crossovers. More than two chromatids can be involved. So far, what we have seen is that this is a state, what we have seen. Okay, only two chromatids cross. This one remains as parental. But this can also be a case. Look, these are 4 chromatids, and these 2 chromatids are crossing over, but then this one is going through this. Even this chromatid is crossing over. Here, all 4 chromatids are crossing over. These 2 chromatids are crossing over, and even these ones are crossing over. So crossing over can give you quite challenging and confusing results. We do this in when we map tetrads, we do in the yeast. Such experiments are really interesting to perform. This is, I already told you, if you are the physical distances are long, or sometimes, you know, recombination gives you no non-parentals or no recombinants, and the reason could be that recombination is taking place near centromeres. So our genes of interest, they are not within the same distance. They are far away from the point where recombination took place, and you will be confused that, you know, you will say zero recombination took place. Same is here. So you have, for example, capital A B, and recombination took place between A and B just at this point, and the gametes are non-recombinant, but actually recombination took place.
Excuse me, sir.
Okay. Yeah.
सर जो पिछली स्लाइड है वो जरा करें.
Yeah.
सर इसमें सारे पेरेंटल्स होंगे ना नेक्स्ट जनरेशन में हमारे पास?
ये सारे वही आ रहे, but recombination did take place.
Yes, sir.
Yeah. Okay.
Sir.
Yeah.
जब हमारे पास रिकॉम्बिनेशन नहीं हो रही, then is there any way to map those genes? क्योंकि 3-point test cross में तो हम through recombinants और double crossovers check कर रहे हैं ना.
नहीं, उसमें नहीं होगा. When we don't get any recombinants, how do we know? Because the physical distance is based on percent recombinants we receive.
Then how do we map those genes?
How do we map those genes? We will bring in another one, another trait, for example, A, B, we will cross A with C, B with C, and we will know that A and B are on the same chromosome. Clear?
Okay. Okay. Yes.
Okay. So, yeah. Now, you should read this slide yourself. This will be helpful. This is self-explanatory. But what I want to show you is that so far we talked about recombination in meiosis. There is almost no or 1,000-fold less recombination in mitosis, during mitosis, than meiosis. Okay. And this was shown in 1936 in Drosophila. So normally mitosis, because homologs, they don't pair, chiasmatas are not formed, and this is very typical for meiosis only. But you know, there is 1 in 1,000 is chance that recombination takes place in mitosis as well. I would like you to leave with the question is, and the question here is, you should go and read what is use of mitotic recombination in fruit flies. Okay. Geneticists, they have very intelligently used mitotic recombination in fruit flies. You should bring an answer with an example. Okay. Give an example, experimental example, not just written text. Okay. Okay. So yeah, this is already, and the second thing is mapping by tetrad analysis. how do you map genes using tetrad analysis? Okay. Okay, now let's come to a very interesting problem Morgan's lab came across, and they solved it, and it led to discovery of sex chromosomes. What Morgan's lab found was a very precious fly, which was white mutant. So what they did, they wanted to study inheritance of white gene. This is just, fly life cycle. You should, as a master student, you should know why fruit flies are useful models in labs. This is T.H. Morgan himself, okay? And by the way, this is the fellow Sturtevant who mapped black and vestigial. He was a sophomore student, BS, second year student. So the next challenge they solved was they had normal red eyes. They crossed normal red eyes with white-eyed male. All they found were normal red eyes. So what do you conclude about white? What do you conclude about white?
White is recessive.
Recessive trait.
White is recessive. Now they took F1 red, crossed them with normal males. Normal males are red, and they get all daughters normal, but half the sons were normal and half were white. If you take normal males and white being recessive, you should not get this picture. Here, white is being designated as recessive. So let's say if we solve it using simple Mendelian genetics, so capital W for red, small w for white, all, it makes sense because this is dominant, all red. You cross now with normal male. Normal male will be like this. We should get all normals. But they did not get all normal. Only daughters were normal and sons were 50-50. What they did, they said, okay, let's change. And they took white female, crossed it with normal male, which is red. What they sorry, what they received, all daughters normal, but all males or sons white, already in F1 here at this stage. This was confusing. So since they paid attention to these flies, they said, okay, here we have used female, here we used normal female, and this one is white female. Just by changing sex of the parent through which white allele is coming, the F1 is different. And they assume that, the gene white is somehow behaving as or sex-linked inheritance it is showing. So could it be we have some chromosomes which are different in males and females? And They went back in literature and they found that in 1905, Nettie and Stevens, under microscope, they had already uh, discovered that, you know, the females, all their chromosomes, so they all chromosomes, they look same. Let's say if this is chromosome 1, it exists in pairs. If this is chromosome 2, it exists in pairs. But there is a chromosome which exists in pairs in females, but not in males. Let's say this is chromosome 1, chromosome 2, and this is now the male. This was female. This is like this. So they named male as heteromorphic pair, this one, where the homologue was missing. There was a much smaller chromosome as compared to the normal chromosome. So this one, where identical pairs of this chromosome existed, in contrast to that, the heteromorphic pair where the homologue was missing. So they named this one as Y, and the one which was present in pair in females, they named them XX. So there was a single X in males and heteromorphic pair in males. So the gametes in females after meiosis, they are homogametic because they all contain X. But in case of males, they are heterogametic because one gamete will be a X, the other will be a Y. So Morgan's lab thought that the Y gene is on X chromosome, most likely. So they said, okay, let's now put it here. We have female, so 2X, and normal means capital W, capital W. Here, Y, capital W, sorry, small w, Y. And in gametes, these chromosomes will go to opposite poles. One will be this, the other will be this. So if we have just like this, small w, Y, and here capital W, capital W, so everybody is normal here because male is contributing small w, which is going to the other X. So the daughters are also normal, sons are also normal. Now, if you take red-eyed females from F1, what are the red-eyed females from F1? They are these ones with 2X. But red-eyed females look like this, small w. Normal male look like this, Y. Okay. Now you can have again the gametes. Male is normal W, Y, and female gametes are capital W, and the other gamete is small w. So what you have now, capital W, capital W, capital W, small w, small w, Y. So half sons, capital W, Y, normal, small w, Y, white. So they solve this, and you can, you know, now answer also why it happened when they used white-eyed females and normal males, why they got all daughters normal and all sons white. So this led to discovery of sex chromosomes. Today in 21st century, we know if you have two X chromosomes, it will be female in Drosophila, female in male. If you get XY, it's male in Drosophila, male in humans. But there are species-specific differences. If you have two XY, it's a female in Drosophila, but a male in humans. And if you have X naught, it's a male in Drosophila, but a female in humans. Why? Because the Y chromosome in humans, it contains a gene named as SRY, which is important for development of maleness traits. And X and Y, they don't contain any homology. No genes on X are similar to anything on Y, and vice versa. No genes on Y are similar to X. So SRY is present near the telomere on human Y chromosomes. That's why even having two X chromosomes, but a Y, you develop as male in humans. And if Y is missing, you develop as a female in humans. So, but in fly, it's different. In fly, there are 4 chromosomes. So X, Y. So this is how they write. Semicolon, chromosome 2, semicolon, chromosome 3. There's 4th chromosome as well, but it's so rudimentary, full of transposable elements. It's silent, so we don't write it, we don't consider it. So what they say, the ratio of X to autosomes, are the chromosomes which are not linked to sex development or sexual traits. So ratio of X to autosomes, if it is 1, fly will develop as female. If it is 0.5, it will develop as male. So here, for example, 1X over 2 autosomes in males, you will develop as, so ratio is 0.5, you will develop as a male. If there are 2 X chromosomes like this, ratio of X to autosomes is 1, so you will develop as female. And when we will, in later part of this course, we'll go into dosage compensation and sex determination. You will see why this ratio influences the sex determination in flies. So you see the discovery nearly 100 years ago. They told us that sex determination is due to these heteromorphic pairs of chromosomes, which are X and Y. And there are genes which reside onto these chromosomes, and they show sex-linked inheritance. They did discover another thing, which was Morgan's lab discovered some exceptions when they crossed white female with red males. So white female is what? You have small w crossed with red males like this. What you should get all females red and all males should be white, because this Y chromosome will combine with either of these two alleles, and the resultant male will be white-eyed color. And this X chromosome, when it will combine with either of these two gametes, it will give you heterozygous, but since white is recessive, so you will get red females, all red, and here all males white. But what Kelvin, who was I believe a postdoc in Morgan's lab, he received 1 out of 2,000 white females and 1 out of 2,000 red males. You expect all males white, all females red, but he was getting exception. And what he proposed, he said that due to defects in meiosis, rare defects in meiosis, and he named them chromosomal non-disjunction. Due to chromosomal non-disjunction, instead of both the homologs going to opposite poles, in meiosis 1, during reductional division, they both end up, they don't separate, they don't go to opposite pole, they go to same pole. They end up in the same gamete, and what happens after meiosis 1, one of the gamete is now capital W, and the other gamete is lacking X chromosome. The other 2 chromosomes are there, but the X chromosome is not present because of the homolog did not separate. They both went to same gamete. And then this goes to second meiosis, and the gametes we have now after second meiosis is this and this. The second meiosis of this mitosis like division will be nullo, no X, because its X went there, so it will be O. The male went normal, X plus, and Y. Now, if this gamete will be fertilized by this one, it will be triple X. So W, capital W, and sorry, mutant, wild type, but it dies, dead. This one with Y, it will be a white-eyed female. Why it's white-eyed? Because there are 2 X chromosomes, and they carry white allele. This one, X, fertilizing this one, wild type allele, nullo, it will be red-eyed male. Why male? Because only 1 X chromosome, the ratio 1/2 autosomes. Okay? And this one is dead. So that's why, due to chromosomal non-disjunction, you get these rare phenotypes. In modern day, what we have learned that due to chromosomal non-disjunction, Down syndrome is chromosome 21 trisomy. So non-disjunction happens at chromosome 21, and we get, Down syndrome in humans. Oh, its time is over. So these are the crosses. You should go back home. I'll upload, and you should solve them yourself. Vestigial short. So you should, this is also part of the assignment. You can see there are rectangles. These are numbers there. Okay. Vestigial short, and then you have cinnabar. So you have to give us physical distance between using these numbers, vestigial, short, and cinnabar. This is a 3-point test cross. We show you, this is the 2-point test, this is dihybrid cross between vestigial and short. And you get these numbers. So you have to give us the frequency of recombination here between vestigial and short, how much map distance apart they are. Then short and cinnabar, how much map distance is between short and cinnabar, and then how they are organized, who is in the middle. And this is the 3-point test cross. Okay. You should also go back and read the genetic problem on this one, mapping on X chromosome. This one. Okay. How genes are mapped on X chromosomes. You already know now the X chromosome. mapping on X chromosome is very simple because you don't need to go through a lot of crosses. Already here in F1 you can look at the recombinant classes. So if there are any questions, please feel free to ask.
सर ये जो लास्ट स्लाइड में आपने म्योटिक नॉन डिस्टिंक्शन की बात की है, ये नॉन डिस्टिंक्शन सिर्फ एग्स में ही होगी, व्हाई दिस कांट अकर इन स्पर्म्स?
It can happen even in the sperms.
Okay.
It can happen in the sperms as well. It's not just in the eggs. Clear?
Yes.
Other questions?
So we need to decide the recitation time with them because I suggested 11 to 12 on Friday, but few of them are busy and not available on Friday, 11 to 12. But I would suggest 9 to 10 or 10 to 11 on Tuesday or Thursday if you all are available. So please ask them.
G. Najma,
सर अगर फ्राइडे को ही किसी टाइम हो जाए?
Najma, can you repeat the time, please?
So 9 to 10 on Tuesday or Thursday, or
G,
even on Friday.
did you send them email?
Yes, sir, I sent them, but 4 or 5 people were busy 11 to 12 on Friday.
So those 4 people should tell us the time then.
Sir, I have a class on Tuesday
Sir,
from
we
9 to
discussed this in
10:15.
WhatsApp group and we agreed upon the early time in Friday.
So if they are busy on 11 to 12, so what other time on Friday they are free?
Sir, I again sent out an email for 9 to 10 on Friday or 10 to 11.